This is a production of Cornell University.
Awesome, cool. Okay, well, today I'm going to be sharing the work of a whole host of people shown here on the screen. But first, I would like to introduce us to the maize classical gene, iodent japonica 1. Now, this gene causes the leaf variegation striping that you see here, and it was cloned in 1992, and it's shown here having 5 exons. Now, if I want to study this gene today, I would go to our maize reference genome browser, where I would promptly see that our V5 annotation version only has 3 exons when I was expecting to see 5. So the question is, what causes this discrepancy? And well, the answer is that gene model annotation is really, really hard. And this is due both to computational errors and biological noise. So if we start by thinking of just the gene models that we know are being translated into proteins, we're going to run into problems right off the bat from alternative splicing. If we expand out and we start looking at the gene models that are being transcribed into mRNA, here we start seeing more problems due to biological noise. So things like pre-spliced mRNAs, fractionation, and duplication. And finally, if we start looking at all annotated gene models, here's where we start to run into more of our bioinformatic type errors. So these are just prediction errors from our software, especially in maize, TE-related chaos in the genome, as well as read mapping errors. Now, if we start shifting our focus from the genome to the protein itself, we know that the cell spends most of its energy on protein. So about 99% of that ATP is going towards generating proteins. And so it makes sense that selection is going to be focusing on what is translated, and we might be able to find those good gene models essentially by looking at which ones are evolutionarily constrained. Now, there's 2 different approaches that we could take here. One is looking at proteins, and the other is looking at more the conservation of our exons. So if we start by looking at proteins, we know that functional proteins have strong evolutionary conservation, both in its structure and folds that persists across all of life. This is really apparent because of AlphaFold 2 as well as ProtTrans. And when we think of ProtTrans, in this example, we have found that functional proteins actually have a grammar that is consistent and learnable across all of life. So what ProtTrans has done here is they've taken over 2 billion proteins across life. They have fed them into a transformer language model, and they basically get out this representation of a protein that is a numerical embedding into 1024 space. And Jingjing in our lab has been able to develop a protein model that accurately predicts if there's a functionally conserved protein in maize. And so what she's done here is she's taken all B73 transcripts, she has run them through ProtTrans and gotten this embedding matrix, and then trained an XGBoost model. And now this model was trained both on the NAM gene classifications, so core is shared across all 26 NAM lines versus non-core shared between 1 and 23. But she also went one step further and she trained on simulated intron retention as well as exon loss, which really added to the robustness of this training set. And as you can see by her model PR and ROC curves here, the AUC is about 0.95. So that means that she's having very, very high precision and accuracy here with her classifications. So we know that there's this, for the gene models that are translated into proteins, there's this grammar that is conserved across all of life. We want to think about this one step up. Into the gene models that are transcribed into mRNA, we can assume that there's this grammar conserved across all the grasses. Now, how this looks is that correct exons we expect are going to have evolutionary support. So say we have 3 different transcripts here for the same gene. If we align them to 4 different species, we can see that this 3rd transcript is the one with the most support. How this looks in maize is we can expand out into the Anthropogoniae, which is the encompassing tribe, and it captures about 1 billion years of evolution. And so as part of the NSF PanAn grant, Taylor Abushan Elder at the Danforth Center has gone out and collected over 800 of these anthropogonate species in a huge collaborative effort by everyone shown here. And we've now assembled over 450 of these anthropogonate genomes, both from short and long reads. So Arun Sitharam from the Hufford Lab at Iowa State did all the long read assemblies, and I've done all the short read assemblies. And as you can see here, we've been able to largely capture our gene space. And so for the remainder of this talk, I'm going to be focusing in only on 33 of our long reads and 58 of our short read assemblies. So this pipeline reelGene, in addition to looking at proteins, can use evolution to predict if a transcript is under selection. For a functional protein using this exact method I just highlighted before, we have our 3 different transcripts. We now can take these anthropogonate assemblies, align them to those transcripts, and see that the third one has the most support. And so how this looks is Zach and Brandon in our lab developed a pipeline to generate a series of multiple sequence alignments to every single maize transcript. So we have 72,000 of these multiple sequence alignments. And then Shanghai was able to find out that the evolutionary constraint of key molecular junctions are in fact predictable by training 4 separate models. And so what he did here, just by looking at the junctions, is he pulled out these different summary statistics for the different types of junctions. So these were our translation start site, our splicing donor site, our splicing acceptor site, and then our translation stop site. He trained this again on our NAM core and non-core designation, but took it one step further and simulated some splice site shuffling. So we have another estimation of errors when it comes to what could be a bad annotation. And then I was able to take those models that Shanghai generated and combine them in addition to some more exon and intron summary statistics and put them into a LSTM machine learning model. Now, if you're not familiar with LSTMs, you can think of them as kind of like a hidden Markov model of the machine learning world. So again, these were trained to generate an exon quality score and predict whether or not we have functionally conserved exons. And now our 2 very different approaches within reelGene both show high levels of precision and accuracy. You'll see our protein and our exon-based models both have AUCs of around 0.95. And so why this is exciting is that reelGene actually allows us to find the real genes in the sense that we can start to tailor things when it's the exon versus the protein-based model. But for the rest of today, I'm just going to be talking about the average of our exon and protein models here. So when we look at how reelGene performs with our NAM classifications, you'll see that only the core and near core genes are largely being predicted as functional. So we're at 89% of our core, while our dispensable and private genes are only at about 8 and 4%. Now I want to highlight these 8 and 4% of genes could actually be sources of adaptation that we could go and look at and potentially help future breeding programs. Now, I want to draw your attention next to this pink area on the screen. These are our classical genes. And so 99% of them are functional. What's one of the ones that's not? Well, it brings us right back to our iodent japonica. And when we look at the reelGene classification, it is predicted as non-functional, which being that the clone gene has 5 exons, it looks like reelGene was correct. And sure enough, if you look back at previous annotation versions, both version 4 and version 3 of B73 have the correct number of exons here. And it's only in version 5 that we see this loss of exons. At this point, you might be wondering, okay, you trained on the core and non-core set. It makes sense you see this beautiful distribution. What about against real data? Well, when we compare it to the maize expression atlas, we find that most genes that are expressed above baseline levels are predicted to be functional, or about 78% meet our 0.5 threshold. And what's even more exciting is that genes that are found in the proteome are largely predicted to be functional. That right now at our 0.5 threshold, we're at 93% are predicted by reelGene to be functional. And we think that those remaining 7% are in fact real genes, but just bad annotations. And so to bring this back full circle, I really want to highlight and emphasize that reelGene is going to be a community resource across species, not just maize. And it's a pipeline for evaluating gene models. So people will be able to take gene model transcripts, feed them into a web server, and then get an output of reelGene scores. Additionally, for all the maize people in the room, we already have scores generated for B73, V5, and we are currently in the process of generating them for the rest of NAM and all prior B73 versions to follow. And finally, I just really want to emphasize again that reelGene is going to allow us to determine the most evolutionarily supported gene models to help out our future research, especially in the case where, say, you have multiple alternative transcripts for a single gene, and you're trying to figure out which one is the best one that we want to use. And so at this point, there's a whole host of people that I need to thank. I also would like to thank my funding sources, give a shout out to Kay McNeary for designing our logo, and I'm more than happy to take any questions. Yeah. So the question for those of you online is, how useful will reelGene be outside of the Anthropogonia? And the answer is it will be useful because we're setting up this entire pipeline so that if you have other species outside of the Anthropogonia, you can follow it and add in your own assemblies, and it'll still take it through the entire training process to the end. So it's basically this framework of how you can compare it to, say, multiple other closely related species. So you can see short reads do work, and you can follow it from there. Yeah, so it's a whole framework that people will be able to use, not just in the Anthropogonia. The TBD. It's in progress right now. Come talk to us if there's a time crunch and we can help you. Yeah, so Jeff's question was, what do you need in order to do this? How many genomes? That actually is a question that we are working on asking. I mean, right now we have, 90 genomes that we threw in there. I am hoping to address this question of how few genomes and how evolutionarily distant do they need to be in order to have that support. I will say, if you want to follow the protein model, you can do that without any other genomes. If you're more interested in the exon-based model, which is really highlighting kind of the conservation of key junctions and other features, I would probably want at least 10 other, just throwing a number out, different genomes, but they can be short read. Basically, all we need to be able to do is align your transcript of interest to those other genomes, and then it can be fed through the pipeline. So they don't need to be annotated. Yeah, so the dispensable and private genes. So those are a classification within the maize NAM population. So the founders are 26 different lines. A private gene is a gene that was found in only one of those 26 lines, while a dispensable gene was found anywhere between 2 and 23 of those lines. So they were basically, specifically found only in that line. Most of them are looking like they're not real genes. But as you can see here, about 4% look to be functional. And that, again, could be a specific, novel source of adaptation you could look at. Yeah, that's a great question. So Anna just asked, how do we balance between evolutionary conservation and a protein that's more newly evolved? My answer to this has been actually the difference in how you weigh the protein model versus the exon model. So that exon model is going to weigh much more heavily how conserved something is versus the protein model. It's basically being trained on, does it have a particular, it's like, are the structures and the folds that kind of occur with that from, basically this representation from protrans, are those basically characteristic of a protein? So I would look more on that side if you're thinking you have a newer, more novel protein. Any other questions? Awesome. Well, thank you.
This has been a production of Cornell University, on the web at cornell.edu.